From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on January 30th, 2025. Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 77th episode of Matters Microbial. Thanks as always for listening, viewing, and sharing the good microbial word. We can always use new listeners, viewers, and guests. Just a few minutes ago, I was down in my little lab checking on some experiments. Now, I simply adore seeing bioluminescent bacteria, and as you can see, it brightened my morning. I recently saw an interesting piece of microbiological art on social media, and it reminded me of some of the art that students have made for me over the years in my classes. Here is a wonderful theme to remember: what does not kill you mutates and tries again. This reminds me of a similar piece of work I had commissioned that illustrates my central philosophy about the microbial world. First evolved, last extinct. Today's topic is quite important to me. For many years, I've been fascinated by bacteria that pursue and consume other bacteria. Sadly, I've lacked the resources to do deep microbial dives in the laboratory on this topic, but I have published a very small amount. That said, it is never far from my heart. I wrote a review article many years ago on this topic and was able to define 4 different types of bacterial predation, as you can see. The cooperative wolf pack of the myxobacteria, the epibiotic strategy of Vampirococcus and similar organisms, the amazing direct invasion approach of Daptobacter, and finally, the periplasmic approach of my beloved Delavibrio. Now, I adore this image because my spouse, Dr. Jennifer Quinn, drew it long ago. The late, great Lynn Margulis illustrated 3 of these strategies much more graphically in another article I will link to in the show notes, as you can see. By the way, no one has been able to cultivate or study Daptobacter, but then, are we really looking? I spent some years studying Delavibrio. Here's an illustration of its biphasic life cycle. The free-living and very swift, that is to say 50 cell lengths per second, attack phase cells impact prey bacteria. They are then able to enter the periplasm, the region between the inner and outer membrane of gram-negative bacteria, and grow at the expense of the prey cell. The invading Delavibrio elongates into a filament, eventually septates, and eventually lyses the prey, releasing progeny Delavibrio to begin the cycle anew. I was first introduced to Delavibrio as an undergraduate at UCLA by the late, great Sidney Rittenberg, an early investigator of this process and organism. He said that Delavibrio created a safe and protected niche inside of the prey cells for growth and reproduction. Later on, I was able to spend a couple of weeks with one of the giants of the field, Dr. Liz Sockett at the University of Nottingham in the UK. Can you imagine me in another country? I commissioned a former Doc Martian of mine, Courtney Sutphin, to make this wonderful needlepoint illustration of the Delavibrio life cycle as a thank you to Dr. Sockett. Delavibrio is fascinating. When I took the microbial diversity course at Woods Hole many years ago, Tom Pitta took this wonderful photograph with me of Delavibrio attaching to prey cells. Here also is a feeding frenzy of Delavibrio attacking E. coli. The arrow points to a poor E. coli cell with 7 or 8 Delavibrio cells attached. I think we can probably cue the theme from Jaws. Now, Delavibrio may sound like a virus, but it's not. It is a bacterium with an active metabolism inside and outside prey cells. It does, however, do something quite interesting. When a single Delavibrio cell begins to infect E. coli locally on a plate, you have what appear to be plaques. I took this photograph when I was in Dr. Sockett's lab, and you can see those plaques. Each plaque represents where one Della-Vibrio cell attacked E. coli in a lawn on the surface of a plate. This is the reason that many people get a little confused about Della-Vibrio versus a bacteriophage. It's a wonderful organism, but often quite challenging to study. Now, organisms like Della-Vibrio are literally everywhere and are seen as increasingly important, including as alternatives to antibiotics. Once again, I will put a surfeit of important links in the show notes. As I've mentioned, my work with Della-Vibrio is actually minor. I will again put links in the show notes to other researchers in the field for your enjoyment and edification. So, it is a real pleasure to introduce Dr. Laura Williams, currently Director of Undergraduate Research at Georgia Institute of Technology and longtime Della-Vibrio researcher, to talk about the wonderful work she has done with undergraduates while she was at Providence College. Welcome to the Quality Quorum for today, Laura. It's really quite wonderful to see you again. It's been a while.
Thank you, Mark. I'm so excited to be here, and I'm delighted to talk with you about Della-Vibrio.
Oh, so much so. And the first thing we have to say is, the term Della-Vibrio, and I've heard it lots of different ways. Some people still say Della-Vibrio, and, I don't argue when they do. I was taught to say Della-Vibrio by Sid Rittenberg, who told me that it stands for curved leech, which kind of makes sense.
Yes, absolutely. And I love that you mentioned Liz Socket. I also got my introduction to the wet lab world of Della-Vibrio in the Socket lab. So her generosity in the field is really wonderful. And that's where I learned, I can't say it with the same wonderful British accent, but that's where I learned to pronounce it as Della-Vibrio.
I've known her for many, many years, and it was really wonderful to spend some time there.
Yes, for me too.
Although, remember, you're not supposed to work on weekends.
They were very serious about tea time, which I really appreciated as a way of establishing good work-life balance. I liked that a lot.
They worked so hard to make me welcome, and that was probably not the easiest job. So let's start talking Della-Vibrio. You had a whole research program at Providence College with undergraduates on it.
I did, yes. I when I talk about this research, one of the things I love to lead with is that everything I show in the talk, all the data, all the figures, all the beautiful pictures, were all through the work of undergraduate students. So from about 2015 until about 2023, I was a associate professor, starting out as an assistant professor, managed to get tenure, yay! Associate professor of biology at the end at Providence College. And Providence College is a small liberal arts school in Providence, Rhode Island. Mark knows that for its H.P. Lovecraft connection, Providence, Rhode Island. But we didn't have graduate students, we didn't have postdocs, and what I had was wonderful, excellent, enthusiastic students who always had the audacity to graduate and go on to do other great things. But while they were with me, and I know you know this too from your work with students, they had excellent ideas, they noticed things in the lab that they would bring to my attention, and they really fueled these projects and the papers that we've produced, which I've been really happy about our ability to produce and publish in peer-reviewed journals. Many of them ASM journals. Thanks, ASM.
I agree completely. So for listeners and viewers, that is how I got to know Laura, is when we would be at ASM and talk to one another. And it was always fun. And I've talked to several of Laura's students, and they were quite wonderful as well, if that matters. But there you go.
Yes. They did excellent jobs presenting their work, and they got to go to quite a few conferences. So it's really wonderful to have that kind of support for students, undergraduate students, to be able to share their work with the professional community. For all of us, we get very focused on what we're doing, and we start to think about things in a narrow way. But as soon as you get out and share your work with other folks that are interested, it really reignites your enthusiasm. So they absolutely responded to that.
It absolutely is true. And, the best moment in undergraduate research is when they're excited to come in the next day depending on the system, sometimes it takes 3 or more.
Yes.
And then want to see what happens next. And there's no substitute for that.
Yes, absolutely. I agree.
So talk about your research program that you had with them.
Yeah, I would love to. So when I arrived at Providence College, I knew that I wanted to get started with predatory bacteria. I had, again, through the wonderful support of ASM, I had a career development grant for postdoctoral women. That's an ASM program, a longstanding ASM program from the generous donor who supported that. And I used that before I got my faculty position. When I was still a postdoc, I used that grant support to visit Liz Sockett's lab. And when I was interviewing for positions, all of the faculty positions that I interviewed for, they were at undergraduate institutions who really emphasized, we want you doing research. What are you going to work on? And I was making a shift from my postdoc work to emphasizing predatory bacteria. And once I got my faculty position, started at Providence, recruited my first group of students, and I sat down with them and I said, okay, folks, we better get our hands on some predatory bacteria. So that was the initial start of this, was asking that first group of students to choose some type of environment that they wanted to go hunting for predatory bacteria. We started at the very beginning.
May I stop you for just a second for
Of
listeners
course.
and viewers? We have a problem in general in microbiology in that when we study a particular species of bacterium that's been used in many labs, I have a saying that the hand of Darwin is on all that lives. And so when we look in the laboratory at laboratory strains, they are fundamentally different than their, quote, wild type originator. And that's because they're evolving to live in the lab.
Yes.
Here's something I don't know that I ever told you, Laura, but Sid Rittenberg, who worked with Dellavibrio bacteriovorus 109J,
Yes.
passaged it twice a week for 20 years until it ever went to the ATCC.
Ooh.
So,
the changes.
right. And
Yes.
so that's why folks, when they went to a different strain of Dellavibrio to study, that's one of the reasons they did that. And so what I really find appealing about what you're saying is you went and looked for them, again, in a somewhat similar way to the way people look for bacteriophages.
Yes.
And with the same issue, if you continually propagated bacteriophages in the lab, they would indeed adapt to those conditions.
Yes. Yes. I mean, bacteria, viruses, microbes are excellent at adapting. It's a virtue of the numbers in their populations. It's a virtue of their fast generation time. How quickly does one cell become 2 cells, so forth. So we were very interested in, and this was a theme from my postdoc research and my grad school research, is that I was very interested in variation. I've always been very interested in natural variation, evolving variation, and I thought the foundation that folks in the predatory bacteria had established by studying the 2 really common lab strains. There's one called HD100. We love it. And then there's another that you mentioned, Mark, already, 109J. And you can learn a ton by taking those well-known strains and kind of dissecting, what does this protein do? What does this protein do? And that's really important. When I saw that foundation was there and that there were groups already really well positioned to advance that work, I thought, well, I love variation. Like, I love learning about variation and understanding variation. And I thought, I feel like there's a space for my group of undergraduate students who are not going to move at the same pace as somebody who has grad students, postdocs, for us to go out and get our hands on some unique things that we could contribute to the field. So we could say, here are new strains. Let's see how they compare. Let's see how they're different. And so over time, my students were able to find, isolate, start to work with and characterize these wonderful predatory bacteria. I had a student who said, I want to go find some from soil. And she did. Mm-hmm. And I had a student say, I want to go find some predatory bacteria in a marine environment. Mm-hmm. And he didn't find Dellavibrio. He found a saltwater adapted predatory bacterium called Halobacteriovorax, which was super tricky to work with. He did a great job with that. And in fact, here at Georgia Tech, recently hired, I have a colleague in biological sciences who's working on Halobacteria vorax and how it relates to corals. So that's a very exciting field. I didn't stick with that because once my student, Brett, graduated, I said, good heavens, I don't think I'm going to be able to keep up with that. He was too good. So we kind of retired Halobacteria vorax after a bit. But the Delavibrio that we got out of soil, we were able to get some other predatory bacteria from freshwater environments. I will say one of the most unique environments that we got predatory bacteria from was there was a student who started in the lab. She was with me in my very early semesters, and she said, I would like to find predatory bacteria in the built environment. And I said, oh, I don't know, there's not a lot of literature about it. I don't know if you're going to find anything, The classic PI move where you go, I don't know. And she said, I don't care, I'm going to try. And I said, sure, okay, fine.
Yeah.
So she thought very carefully about where would you want to look. So it was a good exercise in designing a project, designing a protocol. You would think, what would predatory bacteria need in order to be able to thrive in something that's not soil or not freshwater in a built environment? So she thought that through and she said, well, moisture. We're like, sure, yeah. What are they going to need to do? And she was like, oh, right, they're going to need to eat things. So they're going to need to prey on these bacteria. Where are we most likely to find bacteria? So she started focusing on bathrooms. She started focusing on showers. She told me great stories about like wandering into her, the dorm shower. And people are like, what are you doing with that? Like, why do you have a swab? What's going on? She's like, don't worry, it's science. And so eventually, please go ahead. Yeah.
All I was going to say is it's important, again, for listeners and viewers. I'm not trying to interrupt you, but I want them to understand the way you look for this is so tuned to undergraduates, what are called cures. And it's not that different than people looking for bacteriophages. If you look at this image again of some of those Delavibrio plaques that I mentioned, you have a plate, you have a lawn of the prey bacteria. If there are Delavibrio present, you're going to get those plaques.
Mm-hmm.
The problem is, as you know, if you have bacteriophages, they will also make plaques.
That's true.
So, Laura, how do you tell the difference?
Yeah, so what we were able to do is there's a couple things that you could do to tell the difference. First of all, your bacteriophage plaques are usually going to come up quicker. Bacteriophage, those plaques form, those clearances in the prey lawn that show you that they're eating. For bacteriophage, they're not really eating them because they're viruses. So they come in, they invade the cell, they burst, and then, yeah, that was good timing. And then you get that nice clearance. And phage tend to be quicker than predatory bacteria, so you'll see those plaques come up first. But the other thing that we were able to do is when we pick those plaques, you can put those plaques, resuspend them in some buffer, and then you look for those cells. You can see them. We had a great colleague at Providence College who was excellent with imaging and helped us get set up with a really nice phase contrast microscope setup. And it was very simple for the students to make a wet mount, put the slide on under the scope and say, yep, there they are. You can see these tiny cells attaching to these prey. And you're not going to see that with phage because, as our microbiologists know, our other folks may know, these viruses, these bacteriophage are small. And your predatory bacteria, while small, are still visible under a good phase contrast microscope. And so the students were able to do that, able to see them in real time. They really respond to that too. They love to be able to see, there it is. There's my Delavibrio, there's my isolate. You can actually see these Delavibrio NCO1 in this microscope image. There's 3 panels there, A, B, and C. This is a figure from a paper that we published about it. So that first panel, A, these are just 4 separate pictures that we got with our phase contrast microscope. You could see the really tiny, kind of comma-shaped, those are Delavibrio NCO1 attached to these larger prey cells. Mark mentioned at the beginning of the show that Delavibrio has a biphasic life cycle, so those comma-shaped Delavibrio are going to go into the cell. That's what panel B is showing. You can kind of see there's a little bit of a blob and a smaller blob. That smaller blob is the NCO1 inside that prey cell's periplasm. In panel C, you can see a single Della-Vibrio NCO1 cell. This is at a higher magnification than the other two, which is why it's looking bigger in that picture. So that comma-shaped cell, you can see that shape, and then the long spaghetti is the flagellum, which helps it swim so fast.
And this is so great because, again, this is something that, apart from the visualization technique, is something relatively low-tech. And you're working from nature, so you don't know what you're going to get. And there's something fundamentally fascinating about that to students.
Oh,
I've
definitely.
also, and I think I've mentioned this to you, one of the things that I've seen, and I don't know if this is true with your isolates, is Della-Vibrio plaques tend to continue to grow in diameter over time.
They do.
While phage plaques, they basically stop as soon as the background cells quit growing, is the way that I was told this. And again, you know, when I say that, that's based on probably just a few things, and you may see different things out in nature, which is both frustrating and exciting. But let's be clear on something else. There's a term that your field uses a lot, BALOS. And when people see it, what would you tell them that stands for?
So that stands for, it's BALOS. It stands for Della-Vibrio and like organisms. And that's in a way, it's kind of a lifestyle classification. It's a way for people to say, like, Della-Vibrio is probably the most well-recognized. Doesn't mean it's the most ubiquitous. It doesn't mean it's the most dominant. It just means it's the one we found and the one that we've studied the most in terms of a genus of predatory bacteria. So it gets that place of pride in that acronym. So Della-Vibrio and like organisms is a way of kind of talking about these predatory bacteria. I will admit, and this is perhaps controversial, I will admit that I'm not a huge fan of that acronym. I get it. I understand why it's used. I think it can be very useful. But I'm also somebody who had training, pretty heavy training under my postdoc advisor in molecular evolution. And so for me, I kind of twitch a little bit because the BALOS term can group together phylogenetically, which means kind of evolutionarily disparate lineages. And I think that can sometimes disguise the fact that this predatory lifestyle has evolved independently multiple times. Which is really... a very interesting from one of the reasons that I'm interested in this field is from a bacterial evolution perspective. When you think about the evolution of lifestyles. So why is it that you have a Della-Vibrio lineage, you have this clade of Della-Vibrio, and there's a tree that I have that I've published in a recent paper that shows kind of what I call all the predators. And you can see lots of predatory bacteria in this tree. So different Della-Vibrio, different species of Della-Vibrio. There's that Halobacteria vorax sitting in there that's from the salt water. But also in that tree are relatives, bacterial groups that are related. They're descended from a common ancestor, but they're not predators. So the question then becomes, how does this lifestyle evolve? What are the steps that happened? And it has happened on the bacterial tree multiple times. So I'm always a little uncertain about that term. I like it for reasons, and then for reasons, and it's that. But that's what it is with everything, right? Like anytime you have a term like that, there's different ways to look at it.
You are absolutely right in having these concerns, and I believe it's the fight between lumpers and splitters. Right? I really do. People don't want to put everything in one category. The reason that it doesn't offend me to the extent it offends you is that it's important that some of the Vorax style Della-Vibrio-like organisms are not as closely related as some of the closely related taxa. So it establishes that they're not always the same. But what you're saying is exactly right. This is clearly polyphyletic. It's happened many times, and it can't be from one change. Right, that's something that's really important.
Yeah.
When you become resistant to an antibiotic, that can be from one change. Think about the number of steps for this, and this is why Della-Vibrio is an interesting organism, because it has all those different steps that you see in the life cycle that I talked about.
Yes. Yes.
So you mentioned that you had some of these interesting things. I believe you have a photograph of that drain.
I do, yes. So I'll resume telling you about my wonderful student who took on the built environment. So she picked out places that had moisture, thinking that those would allow bacterial, maybe biofilms, to establish themselves, and that's good food for predators. So she said, I'm just gonna check some drains, right? That's what I'll do. And it turns out that if you walked out of our lab, turned right, walked about 20 feet down the hallway, there was a custodial closet. And this custodial closet had a drain, which I have a picture of. You can see this is your classic, people be dumping mop water in that drain. Sure. And she said, I'm gonna see if there's any predatory bacteria in this drain. Long story short, yes, there was. So, and in fact, we haven't published this yet because I need to sit down and do it. That's also the classic PI problem. But when she was first doing this, she was using a technique called PCR to just look for evidence rather than trying to go through the process of isolating it, because she thought, I just don't even know if it's there. So let me back up a step and use a PCR test to give me some indication of whether or not there might be something there. So her PCR test results for that drain indicated that they were Della-Vibrio. And then she had the audacity to graduate. So I said, okay, we need to reconfirm this, because there's not a lot in the literature, if anything, about Della-Vibrio in the built environment. So let me have another student kind of repeat the process. So I had another student come on. She repeated the PCR test, positive again. So we thought, okay, not only are they there, but they seem to be persistently there. It's not just, oops, it's a fluke and you caught them on a Thursday when the mop water that was dumped in there happens to have them and now it's gone, right? We're getting consistent PCR hits. So consistent evidence that there's Della-Vibrio present. So that second student started the process of trying to isolate them, had a little bit of success, and then also had the audacity to graduate. Gosh, people moving on to their next career stages and doing great things. So I handed that project off to another student, and he managed to isolate 7 different isolates from that drain. Some of them are probably quite close to each other, might be the same things, but we had 7 freezer stocks that he got from that drain. So we started calling them the Drain Della-Vibrio. That was that project, Drain Dello. And he presented some of this work at ASM, and we've sequenced one of the genomes. So if you want to go look it up, he got to name it. His last name was Zappia, so it's called Zap7. So it's Della-Vibrio Zap7.
Please tell me you're spelling drain BDRAIN.
Oh, we should have. We missed that trick, didn't we?
Did you know, by the way, for the people that are doing C phages, that they had to have a rule that the people identifying phages could no longer name them after Nicolas Cage?
Oh, that's great. The Cage phage. That's great. That was one of the things in addition to getting these isolates, my background is also in genomics through, molecular evolution and so forth. So we also wanted to have genotypic characterization in addition to phenotypic characterization. Right. So my lab managed to produce in the phylogenetic tree of all the predators. That tree is built off of Della-Vibrio and other bacteria that have complete genomes. And I'm really proud that my students are really the folks responsible for generating those genomes. So that was always my deal with them, is I said, if you isolate it and you sequence it, you get to name it. So it's exciting for them. They get kind of, installed in the GenBank Hall of Fame, if you will, with their strain name in there, which is always great.
That's a wonderful story. Something to consider, though, and I hope that we'll have some time to talk about applications, because I promise you that's what everyone's into. But I don't. Can we still say geeked out? I geek out about what you find in nature, I'm going to have to probably get this in Latin, that I think that nature still is the best laboratory.
Mm,
And
yes.
it really is. And even when I'm hearing about these drain-associated delevibrios, evolution clearly has taken place there from the time they were first introduced to those that are better able to survive. Perhaps they're
Mm,
stickier to biofilms than others. I just made that up. But the point is, and you've done some experimental evolution yourself, that it's taking place all the time.
absolutely.
And that's what's
Absolutely.
exciting. Yeah.
Yes. Yeah. Yeah. We have, it's always been an ambition of mine to do an experimental evolution project. Folks who are watching this might be familiar with some of the really excellent experimental evolution projects from people who are honestly better at it than I am. But I was really inspired to think about experimental evolution by folks like Rich Lenski and the long-term evolution experiment with E. coli, which is so fantastic. And I understand that he's handed off the care and feeding of that experiment to a new group, which is great because it keeps going. So that's awesome. And so even before I started working with predatory bacteria, I had in mind I need to do an experimental evolution project at some point during my scientific career. It's just too cool not to. But once I got established at Providence, I kind of got handed a really great opportunity by nature and by my students. So one of my students isolated Delivibrio. It's called NC01, and she isolated that from soil. And we noticed when we tested that strain's ability to kill a couple of different types of prey bacteria, we noticed that it was really bad at killing Pseudomonas. It could do it. So this strain could kind of sort of kill Pseudomonas. Like you could see some clearance, you could see some plaques, like you've discussed. You could see, it's not that you can't, but you're just kind of wimpy. And so our big idea was directed evolution. So we thought, okay, what if you take this Delivibrio, who's kind of a wimp at killing Pseudomonas, and you just feed it the same Pseudomonas strain over and over and over and over. And when I explained this, I've presented this project a couple times, and folks who are really sharp on the experimental evolution front always go, but then the prey will co-evolve. And I go, oh no, that's more complicated than what we wanted to do. We just went back to the freezer, got that same Pseudomonas. So the Pseudomonas strain never changed. That Delivibrio ate the same TV dinner day after day after day after day. Maybe it's great. Maybe it was a great and tasty TV dinner, so that's okay. But it basically got the same Pseudomonas prey over multiple generations, over multiple passages, many days. And we wanted to know, could it get better? Could it evolve under that pressure to be better at killing Pseudomonas? And when we got to the end and did those phenotype tests, we were disappointed to learn, no, it did not get better at killing Pseudomonas. And we were a little bit puzzled as to what was happening because we did the genetic sequencing and we could see that there were mutations going to fixation. So meaning that there were genetic changes happening. You really wouldn't see that unless there was some kind of selective pressure. So we thought, okay, well, is the phenotype not changing? What's going on? We had some excellent feedback from some reviewers. We did a few more phenotype tests. And it turns out that it got better at surviving starvation. So we wanted to see it get better at killing a particular type of prey, but essentially what it did instead is got better at going on a diet, I guess, and kind of sort of doing its best at surviving by kind of wimpily eating Pseudomonas, and then otherwise it got much better at being starved. So when we took those evolved Delivibrio and tried to just grow them with no prey, they were great at surviving that. We thought, okay, the exact opposite of what we thought was going to happen.
But this is really important, and I know that you haven't done this. I know that you haven't, but so many people think of microbes like reagents.
Yes,
And
that's true.
and this is kind of relevant. I was really fooled once by a phenotypic strain. I'll tell you about it offline sometime. But what I'm here to say is that microbes have their own agenda and it's not ours.
Oh, that's a
And
yes.
we think we're asking, and so when I say, and boy, this upsets people, I'm not sure we fully understand selection. And what I mean by that is you thought you were selecting for one thing when there were other things that could also respond to that.
Yes, exactly. And I had some really great advice from some real experts in the experimental evolution field, who, it's always one of those things where you're halfway into a project and you go, ooh, I would do this differently. But
Mhm.
they're going to respond, any bacteria, so that includes predatory bacteria, they're going to respond to pressure from lab temperature. We grow them at a specific lab temperature. We grow them in flasks that do this all day. Like, those are very specific conditions that they don't encounter in nature. And they're going to respond to those pressures. They're going to adapt. The population numbers mean that if somebody acquires a mutation that makes them that much better at reproducing quicker, they're going to be the ones that win. So,
Oh,
yeah, I agree
absolutely.
with you.
No, absolutely. I mean, and there's so many. I mean, I have had students select for bacteria that swim faster
Yeah.
on motility plates. And then nowadays we have the ability to take those strains and for about $80 a throw, figure out by comparing the reference genome, what's changed about them. And that's something, it's just, I wish I was about 50 years younger, to be quite honest with you, because
Isn't
we've
it
learned so much.
Isn't it wild what's opened up as a result of sequencing technology becoming cheaper, more available, easier for small labs like mine? If I'd tried to do, just like you said, if I'd tried to do these types of projects 20 years ago at a small undergraduate institution, I don't know that it would have been possible.
No.
But because of all these advances, we were able to pull that genome part into our work, which is really key.
And this is because in many ways, making the kinds of targeted mutations in Vibrio that are really common in lots of different bacteria. I don't want to say the wrong thing. It is more challenging than what you see in many other taxa among the bacteria. Let's
Yes.
just leave it with that. I have been so saddened over the years by this above all things, because things that are easy in one system are more challenging with Vibrio.
Mm-hmm.
And that's why I love and I hate Vibrio.
Yes, it doesn't make things easy.
No.
But if it did, it's the classic, A League of Their Own, Tom Hanks line, right? The hard is what makes it great. If it was easy, everybody would do it.
Well, yes, but I think the other part is, and I take it personally sometimes, because sometimes experiments appear to resist investigation, and I always say I take it personally. And that's being flippant, but it's also true. There's nothing more dangerous than you think you know what's going on.
Oh,
And like you've described a situation where you've discovered it. I have many stories of this, and it all comes back to, when you do a phenotypic screen, you have to be very careful figuring out what you're really asking, because
yeah.
the bacteria don't care what you think.
And I'll tell you, we don't have this published yet, but I'm working on the manuscript. But I have such a great example of that. And it ties into a question that people often ask. So it's a good thing for us to talk about, because I know you have thoughts on it, which is the idea of, can bacteria become resistant to predation? Big topic. We could certainly chew into it. But as an example of getting something you're not expecting, that NCO1 strain that I've mentioned already, that Vibrio strain that came from soil, I had another student working with it, and she was running a very standard protocol: count colonies of prey bacteria, figure out how many of the prey bacteria are surviving attack by the predator. And she came up to my office one day with a plate, and she said, "These look weird. Some of these colonies look weird." And I, again, in classic PI brain. I went, oh, it's you have a contaminant and you just do it again and it's all. And she was very steady, careful, observant. So she went back and she did it again and she got the same thing. She had colonies that looked like your standard E. coli and then colonies that started emerging that looked like, we started calling them squashed because it looked like someone took their thumb and just went on the colony to make it kind of look like a little wacky edges to the colonies. She got it again, and then she got it again. And I said, well, I'm going to do it. And then I got it when I ran that protocol. So it turns out I had a few students work on this project. And through various experiments and various means, what they noticed was in combination of with that NCO1, just that one predator, not another one that we tested, this E. coli strain started to throw off, as it were, resistant colonies. So the squashed colonies, when we picked them and we grew them up and we started that protocol to test whether or not they could be killed over again with just those squashed, they're not killed at all by the NCO1. So we are in the process of trying to figure out what on earth is going on with that. The interesting thing is that the strain that folks work with a lot, that HD100 strain that lots of people know, it can still kill those colonies. Yeah. Doesn't seem to be bothered. So fascinating.
So when I used to teach genetics, I mean, one of the things that I would talk about is the concept of allelism. So if you're working with, say, Drosophila and you have a dark-bodied Drosophila, is there only one way you get to that phenotype?
Yes.
And the answer is no, right? And so in a similar way, I cannot tell you the number of times I've had people tell me that they've figured out how resistance works to Delavibrio. I mean, anytime that you talk about this, and you and I have discussed this, people that aren't familiar with it say, you just do this and it'll come right up. And I'm thinking, so if it's that easy, don't you imagine people have like tried that like me for many years? But what's interesting here is that sometimes we get so fixated on one organism. So HD100, I don't recall its history. It's not like 109J that was passaged for 20 years twice a week. I don't know how long that was passaged, but yours are fresh out of nature.
Yeah.
And it's important for people to note that no one has yet found, okay, now this could be wrong tomorrow. Keep that in mind. That's what's wonderful about science. What the receptor is.
Right.
It is true that Delavibrio tends to go after these gram-negatives. They have to have that periplasm, right? But there are a number of researchers that did the grinding work of changing aspects of E. coli's outer surface one thing at a time to look for, and they've never been able to find something that works for that. But you have found something for your system.
Yeah.
Now, the question is, when you find that, is it applicable to other organisms?
Yeah. And I think what that introduces is interesting perspectives into that broader topic of, can bacteria evolve resistance to predation? So it's a big, chewy topic. It's a fun topic to discuss. The fact that there is variation in bacterial susceptibility to predation tells us that something's going on. It's not a simple either or, yes, no. It's grades, it's degrees, So there's a if anybody's watching this who's like, what am I going to do for my PhD project, friends? Study this. It's so cool.
And it's so interesting because you and I have talked about this specific topic over the years. It's like people who work on this system over a beer, or in my case, a rum beverage, they will share stories that talk about weird things that they've seen with it.
Yes.
Right? And I like that kind of thing, but remember that science doesn't really advance that way. We have to have falsifiable hypotheses.
Yes.
And the reason that this is important, apart from its overall scientific value, and I don't want to minimize that, or as you brilliantly talked about, how important variation is in general, which most microbiologists don't want to think about because it's hard to study. You got to say to yourself, some people have talked about trying to use Vibrio-like organisms as living antibiotics, and
Hmm.
I'll put some links up to that.
Hmm.
But before we do that, we kind of have to know what they're looking for.
Yes.
And you've already talked about a system that you've looked at where even though you had 2 gram negatives, one was preferred over the other.
Hmm.
Why? And no one knows knows that answer yet.
Yeah, and I think it's one of the classic problems in science, philosophy, take your pick, where there's a big broad question, that question of what controls bacterial susceptibility to predation? Can bacteria evolve resistance to predation? And I think when you take that question and couch it in such a yes, no binary term, you're kind of missing this really huge area where there's going to be complexity. There's going to be strain pairings. This predator with this prey is going to lead to this outcome. But you take that predator and put it with a different prey, you get a totally different outcome. So it's a puzzle more than a yes-no question, I think.
Well, and it makes sense. I mean, I remember, because I'm old, I remember this business of how you pick one gene and you hammer it out.
Yeah, yeah.
I don't know if you know this story, but I'm trained as a geneticist, and a seminar I gave very early on, a very famous person who I will name for you offline said that I was a real clear thinker for a microbial ecologist. And I said, well, I'm not a microbial ecologist. And he said, yes, you are. And I'm looking at him and I said, well, no, that's not where my training is. That's not what my interests are. He goes, but you're an ecologist. I said, but I'm not. And by the way, ecologists would agree with me. All right. Just
Oh,
to be
okay.
clear on
They
this.
they don't want you in the club. They
No,
don't.
no, very few people want my insights. Trust me. So the point I'm trying to get at is that you can get very reductionist.
Yes, true.
And
Yeah.
and I want to be clear, and you bring out the good side of me, not the snarky side, is that there's a place for that reductionism, a place for saying, if I change this C to a G in this promoter region, what is the effect of the expression on this one gene? Très coolio, that's wonderful, okay? But is that the question that I want to look at? And the answer is no.
And I
So
and I and I don't this is one of those things where I feel the framework in which we do our science kind of contributes to that. So one of the things I do in my current position is I help our undergraduate students think through how to write project proposals.
yeah,
And anybody who's watching this, who's done a grant proposal, anybody who's done a project proposal knows that what they want to see is specific aims. So define this, test this, and that absolutely makes sense. You need to construct a project in a way where you have some clear questions, some clear outcomes. But what that doesn't give space to is these broader, bigger conceptual things where, like you said, the nature of laying things out in specific games, which is totally necessary, kind of makes you think reductionist, because you need to be able to say, in 3 years of funding, I will be able to achieve blank, you know? So it's harder to say, could you just fund me because I got big ideas about how this whole ecosystem works, you know? So I do think it's tough to balance those two things. And people have strengths in both. Some people are stronger in others. So you kind of bring it all together, and I think it can work.
And I want to bring one further thing up that I think is really relevant to this discussion. You talked about a student who had an unusual result. And so you questioned it, so they did it again, et cetera, et cetera. But it took you some time to accept it.
Oh, yes. Yeah, I went with contamination for longer than I should have.
And I have to tell you, you're quite open-minded. I mean, I know you. So can you imagine a person who wasn't open-minded going with this? And I have a saying for that. I had two experiences with students. So in the early days of what I was doing, I had a gene library of Vibrio. We were looking for adenylate cyclase genes. And I had probably one of the very best research students I ever had who didn't want to pursue it as a career, but here was fabulous. And so I was using a phenotypic screen. and you get a red colony if you have successfully cloned this gene. And she says to me just like this, well, would this also work with the CRP mutation? And I said, well, it ought to. She goes, because I tried it and here's the result.
Oh, wow. That's great.
That's what you're looking for.
Yes.
And because when I was in graduate school and was working with Cynorhizobium meliloti, and I was asked, can you make a Rec A mutant? And this sounds like inside baseball, just bear with me. And then I said, well, if the gene library for this organism is in E. coli that's defective in that gene, can't I just screen in that E. coli? And the person said to me, well, that would never work. That's a silly idea. You shouldn't try it. What I should have done is just done the experiment, because
Just
that is
it.
the way you do it.
Yes, yes. Yeah.
And so
please, no, you finish your thought.
All I was going to say is, that's what Mark Twain used to say. It's not what you know that gets you in trouble as what you think you know and don't.
Yes, that's true.
And it sounds like I'm being critical of other people. I'm not. What I'm trying to say is there's a wealth of niches for the different ways that we look about things to share and engage with. And so I always encourage students, when you say, it's the joke from Isaac Asimov. I don't know if you've heard it, that scientific progress doesn't originate with a damp Greek emerging from a bathtub saying, I found it.
Yeah, that's true.
Archimedes joke.
That's true.
It happens when someone says, looking at something and says, that's weird. And it's true. That is how science goes.
And I think to your point, and I love that you mentioned that the student that you used for this example, you mentioned that they didn't intend to continue with research as a career and did something else. But that's, to me, and I know you know this too, anybody who's worked with undergraduate students on research would know this, is that undergraduate research, doing research as an undergraduate student, and that's why I'm in the position I'm currently in at Georgia Tech, is such a valuable way for students to learn how to think, learn how to reason, learn how to develop confidence in themselves and in their ability to observe and make connections and to synthesize things. And we want people to be able to do those things as doctors, as nurses, as teachers, as people out in industry, as people making business decisions. Undergraduate research, whether you decide to go to graduate school for a PhD or not, is a big part of helping you understand how to solve problems, how to be able to talk about what you found in ways that other people can understand, how to logically present an argument to say, I saw this, this, and this, and I think this. So I'm definitely a true believer in undergraduate research, if no one has been able to tell up to this point.
Well, and I can't agree more. I was trained at UCLA and at Stanford, and the labs that you worked in, what you did, it was a series of jumping through hoops. And if you were lucky enough to work in a research lab, then you start to understand how things work. But not everybody has that opportunity. This is why the rise of cures, course-based undergraduate research experiences, are so important. For example, when I've been working a little bit on the Tiny Earth program with my first-year students, I'm no longer teaching that course, time passes, and a student who is no way interested in biology would find an organism that's clearly making some kind of antimicrobial that works on 4 or 5 different possible relatives of pathogens. They did that. And they found it in a sample that they collected. There's no substitute for that.
None. Totally agree. Yes, I agree.
It is true that sometimes, and we've got to be fair about this, sometimes the cures are all about providing data for a PI's research program. And there's a place for that too. But what I was doing was making sure that from wherever the students came, they understood the goal of what they were doing, that they made it happen. And I think it's wonderful.
Yeah, and in my current position at Tech as director of undergraduate research, so I've moved out of my faculty position. I now live in Georgia instead of Rhode Island. It's a world of changes. So since July 2023, I've been in this position. And one of the things I really focused on when I was interviewing, but also since I've been here, is course-based undergraduate research experiences, cures. And it's so apparent that when you, and I taught with Tiny Earth as well, and when you start the course by saying, here's what we're going to do, here's what we're going to learn that nobody knows right now, so you're going to get discovery, and here's who cares about it. So you get relevance. And when I said those three things to students in my micro class, when I said, here's what we're going to do, we're going to isolate novel antibiotic producing bacteria from soil because they're all out there, I guarantee you. And we're going to learn something that nobody knows yet. And then there's a whole community of people, including some folks at Wisconsin-Madison, waiting for your isolates so they can see if they can turn them into new drugs. It was, students were like, it's game time, let's go. They loved it.
Well, and a really good example, too, is that the person who helped discover Thermus aquaticus, who's the source of Taq polymerase that originally made PCR possible, that was an undergraduate student with, by the way, the coolest name in science, Hudson Freeze.
I don't know that I knew that detail. That is an excellent name.
It is a supervillain's name, let's be clear.
Dr. Freeze.
Dr. Freeze, and he is called Dr. Freeze, is a cancer biologist in San Diego, but he did this as an undergraduate in a summer project with Tom Brock. And I don't bring this up to be flippant. I bring it up that so many students think that they can't contribute to that because they don't have the machine that goes ping, as Monty Python would put it. The most important piece of equipment is here.
Mm-hmm. Yes. And it's a big part of what we want students to do in college. There's lots of discussions right now about why are students going to college? What are they getting out of it? What's the value? But I can tell you from the time that students start as a first year, and you could see this clearly where I was at Providence, and I can see it now too at Tech. They start as a first year kind of more traditionally as a student. There's a tendency to be like, okay, you're a passive receiver of knowledge. We're going to tell you all the things that everybody else has discovered. And you sit there and you absorb it and you spit it out in exams. But they go from that as a first year to graduating as somebody who, through these experiences, through doing research, through cures, contributes to their professional community. They are a member of that professional community. They are generating knowledge. They are contributing to advancing the technology. And that's what they want. They want to generate new knowledge. They want to contribute. They want to make an impact to a person. This generation of students coming to college wants to do something meaningful. So they really respond to things like cures and undergraduate research.
Absolutely. And this is one size does not fit all.
Right.
And we make that mistake all the time. And, you have to meet them where they are.
Yes.
And one final thing, because I know you're such a wonderful educator. I don't know if you agree with this, and it's fine if you don't. I call it the 80-10-10 rule. 10% of our students are going to do great no matter what we do. 10% are going to have real issues no matter what we do. And we like that first 10% because it pets our delicate little egos. And we worry about the other 10% because we want to be able to help them somehow. But remember, they would be in that situation regardless of pretty much anything you do. We're ignoring that 80%. And I think that that's where the cures are particularly important.
Yeah.
And that's been my experience. It's been yours. And it needs to be highlit.
Yeah. And I think too, one of the things is individual faculty have so much going on. Their bandwidth is limited, their time is limited. I admire tremendously folks who take on, these above and beyond type efforts where they turn a course into a cure, or they start to build in things like community-based learning. To do that right takes time. And I have a lot of respect for those folks. And I did think when I moved into this position, because now, as my husband likes to joke, I am now on the dark side. I am a higher education administrator. He's like, ooh, you went to the dark side, huh? It's like, okay. But to me, the role of somebody in a position like mine is to set up the support, set up the infrastructure for both the faculty and the students. So it's our job to try to think about the 10%, the 80%, and the 10% so that the faculty can try to get the courses delivered, the experiences delivered, so that we can say, we've got this in place. So if you have students who are really in crisis, we've got this in place. You don't have to do it yourself. You can send them to these folks. These folks will help you. And that's one of the things that's been a shift in thinking for me. It's very different from being a faculty member. I always joke that faculty, I mean, I don't know if the faculty watching this will agree, but I always joke that as faculty, we kind of see ourselves as independent contractors. You know, like the idea that my department chair would have said this too. The idea that my department chair was my boss was kind of laughable a little bit. Like even my department chair would have been like, not your boss. But once you kind of get into the administrative side, I think that's where you're supposed to be responding to these things, to say, how do we make sure faculty are able to do what they want to do? How do we make sure they have the support? How do we try to make things less burdensome? Like I'm the person in my office going, no, you don't need this paperwork. No, we don't need to require this. No, we don't need to be doing all these forms. Like these people have no time.
Don't worry. They will take you to the top of the academic pyramid and bring the obsidian knife and remove that from your brain soon enough. Share that with your husband. That's his view too. My wife is an administrator now as well. So
yes,
I speak with authority on this.
Yes,
well, listen, yes. It's been a fabulous time chatting with you. I'm afraid I got too excited. These things happen, but it's a topic that I've long been interested in. I'm so delighted to have talked about this with you, not only your research, which I knew pretty well, but also your thoughts about the future of that research and about your new position. And I hope to have you back at some point and we can talk more.
Yes, I would love that. This was really excellent. I don't get many chances to take a block of time and think about science. So I did work in administrative stuff. So there I kind of did my job the way I'm supposed to. But thank you, Mark. I really appreciate it.
There's a box to be checked off and you can put that in your file.
That's right. Thank you so much. I know how that works
too. My very best wishes to everybody there. I have to get out there.
Yes, you absolutely do. There is a thriving and wonderful community of microbiologists here at Georgia Tech.
But I'm an ecologist, so I don't know that they'd invite me, like I told you. So thank you so much for your time. Best wishes to all.
Thank you.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with particularly epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Laura Williams is Director of Undergraduate Research at Georgia Institute of Technology in Atlanta, Georgia. Dr. Williams was formerly Associate Professor of Biology at the lovely Providence College in Providence, Rhode Island. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.